Good morning. Welcome back to CS372, Artificial Intelligence for Disease Diagnosis and Information Recommendations. And the last time, Dr. Melissa Cole gave us a lecture on cancer causes and the diagnosis. And today we welcome Melissa to come back to give us the second lecture on cancer, focusing on cancer treatment, including radiology, surgery, chemotherapy, and targeted therapy and immunotherapy. Let's welcome Melissa.
Yeah, awesome. Welcome back, everyone. So yeah, we will be continuing with sort of the second half of what we spoke about last week. So last week, we really thought about how is cancer caused? How do we diagnose it? And so really looking at what's going on in our cells and in our DNA that leads to cancer, and thinking about the different features of a cancer cell and maybe how that might contribute to what we're looking for in cancer, like diagnosing it as an earlier stage versus a later stage, as a more aggressive cancer or a less aggressive cancer. And ultimately, I think it was clear from that conversation that our goal when we were diagnosing is ultimately that might guide better treatment. So we're going to continue that conversation today. So what I'm planning on talking about today is giving you a sense of a lot of the existing treatments for cancer. So we have what I guess I'd call traditional cancer treatments, which are probably the ones you've heard about or seen in like popular media whenever cancer comes up, whether it be in a TV show or a book. There's typical types of cancer treatment that we've been using for many, many years. I'll talk about some more recent cancer therapies. And then we'll talk a bit about what the issue is with those therapies. So what is the issue with the therapies that we usually use to treat cancer? How is it that cancers become resistant to treatment? And what are some possible workarounds for that? And then I'll again talk about machine learning applications. So we'll think about how could machine learning help with discovering new drugs, or discovering better drugs, or discovering more effective combinations of drugs. And then I will talk a bit about immunotherapy. I know immunotherapy is a big area of interest for a lot of us. I think compared to a lot of recent innovations with cancer treatment, and immunotherapy is sort of just like a different approach. And I think before it was kind of like, you can imagine we developed drug A and we were kind of incrementally improving on drug A, or just slightly, pairing drug A with drug B to slightly improve the efficacy. But immunotherapy is like, let's just take a new approach. And in some ways that maybe will be more effective, especially for certain patients. So I'll talk a bit about immunotherapy, and then I'll just touch on areas of work and maybe areas that connect to careers you might be interested in. And I did try to consciously leave more time. So I think I have fewer slides and fewer things to touch on than last week. So hopefully that'll give space if we want to have a more open conversation, and you can ask either about research or about sort of transferable skills or ideas if you're interested in like the healthcare context or cancer biology context. So that's the plan for today. Let me get started. First off, before we kind of jump right into talking about cancer treatments, I did want to talk a little bit about, again, I think last time I gave an example of like different types of machine learning approaches. So like supervised versus unsupervised learning, how they relate to cancer, like what are the types of problems we think about in the context of cancer when we're thinking about using clustering or using different types of modeling, like classification, regression. So those are kind of like paradigms for like types of problems you'd solve and how they relate to cancer. But I also want to talk a little bit methodology wise. One thing that comes up a lot in cancer is this, it comes up in machine learning, is this trade-off between very complex and accurate models, but they become models that you can't really scrutinize. Like you don't know what's happening under the hood. It's just sort of a black box. You feed in your inputs and you might get really accurate outputs, whether that be a regression or a classification, but you don't know what's happening per se in the model. And so this actually comes up a lot when we are working as researchers in terms of how do we choose which model to use? And it really kind of where we fall on the spectrum of preferring accuracy and giving up the interpretability versus preferring interpretability and giving up accuracy kind of depends ultimately on our goal as researchers. So for example, as a cancer researcher, if your goal is really to try to reveal how cancer works. So this comes up a lot in sort of basic research, thinking about how is the cancer functioning? Does the cancer function? So in that sense, when we're trying to model a process, we're really interested in what are the important features, what is leading to one outcome over another. And so it's not just enough if your goal is to understand the cancer to say, oh yeah, you as a patient have the aggressive cancer and you as a patient have the less aggressive cancer. We want to know what's leading to that distinguishing, that distinction between the two. Is there a really important feature in our cells? Is it a really important mutation? Like we want to be able to pull the model apart and reveal what that feature is. And so a lot of like, how does cancer work kind of problems, we tend towards these more simple models. But then of course, I think there is the clinical application, which kind of falls up here, which is where there are definitely people whose ultimate goal is, I have real life patients, humans dealing with cancer. I just want to be able to have high certainty, high accuracy in telling them, oh, you have this type of cancer. This is the best treatment regimen for you. And so when you're thinking about how to approach cancer research, really knowing, is your goal to understand cancer better, or is your goal just to optimize something so that you maximize the good for your patients? And that's something that we struggle with. We know that cancer is so complicated that a lot of the more simple models are definitely oversimplifications of how cancer works, but ultimately our goal might be just trying to pull apart, like, what are the mutations that contribute to cancer. And those kinds of things would be lost if we used really complicated models that make it really hard to figure out what's going on. So this is a trade-off that we struggle a lot with, and it just kind of depends as a researcher, like, what are you ultimately trying to contribute to? Like giving doctors the best tool and giving the best tool for patients, or just like furthering our understanding of the cancer. All right. So let's return to our case study from last week. So you might remember that last week what we talked about was our neighbor Sasha has been diagnosed with stage 4 pancreatic cancer. So you both know the stage of the cancer, and you also know the type of the cancer. It originated in the pancreas. If you remember last time, one thing we noticed was that and what contributed to the diagnosis of stage 4 was that it was affecting the kidneys as well. So it is a cancer that has metastasized, or it has spread to another organ. And so what I want you to just spend a quiet moment reflecting on, and you can share your ideas in the chat, is what treatment options do you know for cancer, and which ones do you think would work or not work in this scenario? So you can just sort of recall what you've heard about for cancer, and then think about which ones do you think would be used here and might help versus ones that might not help. So just take a quiet moment to think about that. And again, share your answers in chat. Yeah, so I see people mentioning chemotherapy and surgery. So I'm seeing comments about chemotherapy and how effective it might be. Again, there's a lot of trade-offs with chemotherapy itself. Chemotherapy is a very harsh treatment, which we'll talk about in a second. And then surgery, thinking about surgery, you would need to know where the tumor is, and you might clearly know where the tumor is with regards to the pancreas and maybe even the kidney. But one thing that's never really clear with a cancer that is late stage and that has spread is it somewhere else and we just don't know it yet. And so that makes surgery really difficult. And you can imagine with a very sick patient, you don't want to keep putting them under the stress of surgery. Awesome. Thank you for your ideas. So yes, I think really thinking carefully about what will work at this stage in the game, knowing that the patient is probably sick and will only get sicker, knowing that the cancer isn't localized, it has spread, all of those are kind of information that will inform our decision. And generally speaking, when you're asking if you're faced with a patient who has cancer and the first question we often ask is staging, because that tells us a little bit about how bad it is, how far along it is, what are their chances. But really from the treatment perspective, it really guides what we're going to do about it. And so this example here, I believe this is taken from colon cancer. This mapping is really showing you like, what would you do if cancer was early versus what would you do if cancer is late stage. And so what you can actually kind of see from here is that certain things become more of an option at different stages and versus other things sort of fade out. And so an example I'll give here is that when colon cancer is developing, it's just usually a small growth, which we'll call a polyp. And if you find that polyp early, you just cut the polyp out. Like usually it's kind of a benign growth. I guess the way you could visualize it is kind of like the way you'd picture like a wart on the surface of your skin. Just imagine that on the surface of the lining of your gut. Like that's how we think of a polyp. It's like it's a abnormal growth, but maybe it hasn't had the chance to like really be invasive, like in its own context or beyond its own context to other organs. And so you just cut it out. And so that's definitely the main option that you would pursue early on. There's really no reason at that point to use chemotherapy or any of these additional lines of treatment. You just cut it out. Versus, as what you can see in this diagram, sort of as you go further along, this is the surgery option. You can see it kind of just fades out here, because when you're dealing with metastasized cancer, again, maybe you could cut out the main tumor or the tumors that you've identified. But again, it's a trade-off where if the cancer is metastasized, generally speaking, surgery at that stage is not going to cure the patient. It's more like, is it worth it to maybe alleviate their symptoms, like to relieve pain? So if there's a giant tumor and you know that tumor is metastasized to somewhere else, the only reason why you might consider surgery is if it sort of, if that in combination with other things might eliminate the entire cancer and everywhere it's spread, or if the patient is suffering a lot, you might consider cutting out the tumor, just because that tumor is probably still a huge burden and leading to lots of effects on their health. But you can see in terms of really thinking about it as curative, at that point, surgery is no longer curative, so it kind of fades out as an option. Radiation is similar. We'll talk about radiation in a second, but radiation generally tends to be something that kind of like surgery, you kind of want to point to a specific space and say, treat here. And so again, radiation, you don't want to radiate the whole body, you're trying to target the radiation. And we'll talk about why we use radiation in a second. And then in terms of options that become more relevant as we get into later stages, chemotherapy is one of them. So you can see chemotherapy sort of fades in as we go further along in staging. And then there are some more targeted therapies that we use, especially for aggressive and late stage cancer. And we'll talk about these different targeted therapies here compared to the general chemotherapy, which is probably the most common treatment that you've heard about. So this anti-angiogenic therapy and this anti-EGFR therapy, those are targeted therapies, and those are ones that might be used for more than one type of cancer, but you might not have heard about it in any cancer context. Like these these specific ones listed here aren't ones that you see used in every type of cancer. You might have heard about them in the context of lung cancer or maybe brain cancer, but you don't hear them with literally every type of cancer. And that's because there is some specificity involved. All right, so let's talk a little bit about how these treatments work. So what I can just say is that in general, most classic cancer treatments, the way they work, and this might seem obvious, but we're going to kind of dig into this a little more, is that the way they work is they're either physically removing the tumor and the tumor cells, which is the case for surgery, or they're inducing what we call cytotoxic effects on the cancer cell. So surgery over here, you just cut it out. You cut out the tissue. There's definitely a challenge where you're trying to think of the margins of like how big is this tumor? How much will this affect the patient negatively if I've cut out such a big part of their body? And you don't want to like miss some by accident. So that's definitely the struggle with surgery. Versus the cytotoxic effect really is taking advantage of, we talked a bit about cells and DNA, and the goal of cytotoxic effects is that you're killing cancer cells specifically and trying not to harm normal healthy cells, which is challenging, as we'll talk about in a second. But basically, we're either cutting out and removing the tumor tissue from the body, or we're trying to induce cell death in the tumor cells specifically. So how do we induce cell death? So radiation is one way. As I said, radiation is something that generally has to be, you want it to be targeted. You don't want to sort of be introducing radiation to healthy parts of the body. Radiation has been talked about in terms of, we use it for diagnostics, like we take an X-ray and we might see something that looks abnormal, right? So we both use it on the end of, when we first have a patient coming into the clinic, we might use this to diagnose that there's something wrong and lead to the diagnosis of cancer. But we're also using it on the treatment end, where we're taking this radiation and in really high concentrated, strong doses, we aim it at where we think the tumor is. And when radiation hits the tumor, it basically is inducing damage to the DNA. So I think I showed last time the example of a mutagen, like a cancer-causing agent, was radiation. So when your skin, for example, is exposed to a lot of sun, it gets a lot of UV radiation. And especially if you are prone to getting a lot of sunburns, that's sort of representing that your cells in your skin are getting a lot of radiation dosages, and it's probably actually hurting those cells. Because like basically the radiation hits your DNA and can cause that DNA to mutate. When that happens in our body, our cells will usually spot that there's something wrong and try to repair the damage. But if you have an incredibly high dosage of radiation all at once, generally speaking, there'll be so much damage to the DNA that it's kind of hard for the cells to repair, and they actually will tend to make the decision to basically kill a compromised cell. Like if there's a cell that's highly mutated, it is beyond repair, that cell will, generally speaking, die. It won't be able to recover from that much damage. So here you can just imagine we're taking high doses of radiation, we aim it at where we think the tumor is, and we sort of shoot the radiation there, induce mass amounts of DNA damage in those cancer cells, and those cancer cells, generally speaking, might not be able to recover from it. So of course, again, that becomes less of an option if the tumor isn't localized, because we don't want to just radiate the whole patient, right? If the cancer cells have sort of spread everywhere and we don't know where we are, we don't want to introduce that radiation anywhere, right? Because that's going to be harming and mutating normal cells too if we don't focus it. So again, that becomes less of an option in late stage. If there are any questions, I have the chat open, but you can also click the raise hand button, and I'm happy to answer questions as I go. You don't have to just wait to the end. I'll keep an eye if I see any. All right, and let's talk about chemotherapy. Chemotherapy is actually pretty similar. It's just the delivery mechanism is very different. I'll say, generally speaking, a lot of chemotherapies actually mess with our DNA, mess with DNA's ability to replicate itself. So if a cell is dividing, it needs to make a second copy of its DNA so that the two resulting daughter cells both have their own copy of DNA. And so if that process is messed with, then those cells just don't divide, or they divide and they run into issues, and that might either stall them in the middle of dividing, or they might die because they can't recover. They're kind of like stuck and they kind of can't fix it. So at some point they will, those cells will just die because they can't resolve the issue. And then some of these chemotherapies will mess with cell cycle, which again is that process of cells dividing. So there's a particular drug, for example, that messes with the ability of the cells to sort of partition themselves, like they're trying to move things around so that they can split into two cells. And basically the physical things that help with the manipulations and pulling things apart actually just stops working. And so they're just kind of frozen in place. So there's lots of different chemotherapies that basically mess with these processes. They mess with our DNA, they mess with making more DNA, they mess with cells dividing. And as that kind of implies, that's kind of a problem, because healthy cells have DNA too. Healthy cells replicate themselves too. And so this really hints at why chemotherapy has so many side effects, because when you're introducing a drug, and I'm just showing a drug here, this is Cisplatin specifically. It's a very commonly used chemotherapy. It's used with breast cancer, probably also lung cancer. Lots of different cancers use cisplatins or related drugs like carboplatin, which is just a different chemical formulation. But all of these drugs basically go in and stick to your DNA, and that causes DNA damage. So you can see here with this little chemical here, it's going in and it's basically sticking itself physically, like it's chemically reacting with our DNA. And this, in the mind of the cell, is damage. It's basically messing with your DNA structure. And so if we were trying to read this DNA or trying to copy this DNA, we would run into issues because the chemical structure of the DNA is different. And so this is particularly potent for cancer cells. Cancer cells are kind of, how do I describe them? They're kind of like, cancer cells may come across as very powerful in terms of all these additional abilities, and the fact that they sort of conquer the environment they're in. Like, they basically can kind of hijack certain processes, steal blood vessels, steal resources, but they kind of also just like rule over the normal cells around them, because the normal cells around them obey all these rules and the cancer cells obey no rules. So they seem very powerful, but cancer cells also kind of are skirting the line where they themselves are so warped and mutated. In some ways, they actually can be very fragile. And so when you're giving them DNA damage, sometimes they're like, yes, more mutations, like this makes me more powerful, but then too many, and they actually don't have the normal means of resolving them. And so they can actually be more sensitive to things that mess with cell cycle or mess with their DNA, because they've lost the means to repair those. So they were kind of super powerful, and then they got too much of it, and then they just sort of give up more easily because they just can't fix it. But yeah, this is also a problem for our healthy cells too, having this sort of compound go around and bind to DNA, and especially mess with DNA in cells that divide. That's what leads to a lot of the side effects that we see with chemotherapy. When we're thinking about treating patients with chemotherapy, it's basically this really careful balance that's sensitive to the patient. It's sensitive to the patient's age and like coexisting health conditions, right? If a patient has cancer, but they also have, they're also older, or maybe they also have diabetes, they have other conditions. When we're trying to figure out how much chemotherapy to give them, there's sort of this instinct to give tons of chemotherapy, right? Like they're fighting this battle with cancer, and we're trying to kill as much of the cancer cells as possible. But basically, as we ramp up the dosage of the chemo, we just get more and more negative side effects. And there are some patients where literally, if we just kept giving them chemo, the chemo would end up killing them, because the chemo does things like it harms the lining of their gut. It harms their ability to produce blood cells, so they become very anemic. All of these things are like symptoms that you may have heard cancer patients experience. And that's being caused by the chemo. Like, of course, the cancer is hurting them and is causing issues, but a lot of the sort of hair loss, nausea and vomiting, anemia, skin issues, like a lot of those are coming from actually the chemo. And so we're trying to give them high dosages to be an effective treatment against the cancer, but then we often have to hold back because of what the patient can tolerate, right? Both in terms of like quality of life tolerate, but also in terms of literally too much could actually end up killing them if it's just, you know, constantly eroding at their gut and constantly eroding at their ability to produce blood cells. If there are any questions, just let me know. Let's talk a little bit about newer treatments. So we talked about surgery, radiation, chemotherapy. Let's talk about targeted therapy, right? Because I think one thing we can realize with chemo is that it's just sort of this, it's sort of this like shotgun spray, like it's a very aggressive treatment and it kind of just hits. There's all of this collateral damage that's going on because of the nature of chemo. It's like, to kill cancer, we're going to target DNA. It's like, well, that's a feature shared with all cells. So that's kind of a problematic approach. So targeted therapy is trying to be a little more like, you know, narrow the scope, look for a specific feature that maybe is more unique to cancer cells, or again, is something that just doesn't hit literally every cell in the body. And so targeted therapy, the real goal there is that by being more specific, you just limit the amount of you know, side effects that the patient has to deal with, and maybe you can then up the dosage because the side effects are more tolerable, it's less harmful to the body, and again, you can then be more aggressive towards the cancer itself. Yeah, in terms of thinking how to personalize chemo, I would say that there are cases where you can imagine that when making, I mean, it's such a tough question when you're like, how much chemo do I give this patient, right? And you're taking into account all of these features about their current health, but you're trying to make a prediction, right? Because you don't want to be in the case where you went real easy on the chemo and it actually wasn't super effective and the patient's cancer just kept growing, kept spreading, right? Like that is such a terrible position to be in, to realize, oh, I should have been more aggressive because the cancer was continuing to get worse in the meantime. So in that sense, having more data to influence that choice, to be like, okay, because of everything we know about you, whether that be the sequencing, the genetics of your cancer, or some other data, just like knowing your circumstances and other patients who are like you, if that could tell us, yes, it's really difficult, but we just have to be more aggressive upfront. Like, we know that if we don't, chances are very likely that we might as well not treat at all. I think that kind of information could be very useful, because currently, yeah, it's a regretful position to be in if you kind of ease up on the chemo, but it just ultimately isn't helping the patient at all, right? Like, you don't want to have them go through the stress of chemo and not have it add additional, you know, quality time in their life. I will speak a bit in general about how chemo doesn't tend to be curative for most patients. Usually the conversation that's being had is how much can we add to their life? Like, chances are, if it's a very late stage cancer, they will probably die to that cancer, but you're hoping that you can add several months or maybe even a couple of years to their remaining life by treating with chemo. So that's kind of the trade-off, is like, you don't want to give them chemo and have them suffer and not actually add any time to their life. So if we had the data to know, based on this patient, based on the features of their tumor, based on their patient level features, we know we should be more aggressive or less aggressive. Like, don't make them suffer chemo if it's not going to help them in the end. That's the kind of decisions you might want to make. All right, so let's talk a little bit about targeted therapy. So targeted therapy is something that's thrown around a lot. It actually isn't super new, in the sense of, we discovered some really early targeted therapies just based on our understanding of normal human biology. And that helped us discover some particularly useful targets. But since those early discoveries, a lot of the targeted therapies we've discovered have to be more involved. Like, we've had to turn to big data to discover new targets. So the classical targets for targeted therapy were actually things that are related to hormones and sexual development. So if you think about what hormones do in like the typical human body, whether biologically male or biologically female, the way a lot of sex hormones work is that when they sort of kick into gear during puberty, they enact a ton of changes in the body, right? When these hormones sort of ramp up and are being expressed more and they're moving through the body in people's teenage years, it leads to the development of, the sex organs, but it's basically switching on all these pathways for a lot of tissue and cells to develop, right? It's basically saying, okay, time to create these new organs or develop these organs, and it basically ramps up growth and it ramps up cell division in those tissues. And so our bodies have this sort of natural programmed way by which, obviously as we grow older, we just like grow bigger and taller, but also that's like a very key moment in our species development where we just suddenly will develop new tissues and grow new tissues. And so there are lots of cancers that arise in sex organs that sort of hijack that normal process. So for example, in breast cancer, in normal woman development, it's like when the hormones start, that actually kickstarts the development of the breast. So like breast tissue starts to divide and proliferate. And so when you have a breast cancer, it's actually using that pathway to sort of tell itself to keep growing and dividing. So it's like what happened in puberty to lead to growth of that tissue, cancer is using that pathway to just tell itself continuously to grow and to divide. And so that's a very precise pathway that's specific to cells that relate to the sex organs. It's not true of all cells, right? Like not all cells in your body are using, for example, estrogen in that way. And so when someone realized, hey, these breast cancer tumors seem to be expressing a lot of estrogen receptor, like they're really responsive to this hormone, that made them realize, oh, if we block the signaling, the messaging through this hormone receptor, so that it stops basically picking up on any amount of estrogen and telling itself, divide forever, keep dividing. If we block that, it is something that the tumor is very reliant on. Like it really uses that hormone receptor to power its ability to keep dividing. But that's not true of all of the healthy cells. It's true of some of the healthy cells. Like there are some normal cells, especially in a woman's body, that do respond to estrogen. But it's not true of literally every cell in her body, right? Literally every cell in her body has DNA. So if we target DNA, that's a huge problem. But if we target this estrogen receptor, it's just more specific. The eligible population is a lot smaller of who would be affected. And so that's what they basically did, was they created what we call these estrogen blockers. An example of that is Tamoxifen. So Tamoxifen is something that kind of looks like estrogen. And when it's flowing through someone's body, if someone's undergoing treatment for breast cancer and they know that they have a cancer that is reliant on estrogen, which is not true of every breast cancer, but if they test and they know, hey, you have breast cancer and you specifically have estrogen dependent breast cancer, it has a large amount of this estrogen receptor because it mutated in a way to have tons of it. If they identify that about the tumor, then they know that they're a good candidate for this specific treatment, this targeted therapy. And so you add the estrogen mimic, this tamoxifen, it'll bind to the estrogen receptor, but it doesn't actually send the signal. It basically blocks the ability of that receptor to respond to normal estrogen because it's kind of sitting in its spot and blocking their ability to talk to each other. And so generally speaking, if there's a cancer, again, that's really reliant on this estrogen signaling, it can no longer send those signals, and chances are its ability to divide and grow will be stalled. And in some cases, without that kind of continuous encouragement, the cells might also die. This is maybe one thing that isn't super apparent about cells, but if you ever go on to study and research cell biology, we grow lots of cells in cultures and dishes, but that's not actually very typical of normal cells. Generally speaking, if you extract cells from a person and put them in a dish, they won't grow because they don't have the environment they expect. And the environment they expect is both the interactions with their neighboring cells who like tell them reassuring things, but there's also like signals that our bodies produce that sort of flow everywhere that help reassure our cells. Basically, I don't know how to describe this other than saying like our cells get really lonely. Like if you just extract a cell and put it in a dish without all of the messaging that it expects and all of the communication with its neighbors, those cells will actually die because they don't have that constant messaging around them. So cancer cells can be similar, where you sort of starve the messaging that they're expecting and they'll actually die without that messaging. And so we have used this very effectively, again, specifically in patients who use estrogen receptor. So if we test their tumor and we're like, oh, your tumor is dependent on estrogen, it has a lot of this receptor being expressed, those patients will do very well with tamoxifen, with this particular therapy. If they don't express a lot of this estrogen receptor, their tumor isn't using the estrogen pathway. And so the treatment doesn't make any difference to them. And so at the diagnosis stage, you would diagnose someone, for example, a woman with breast cancer, but you would also then genotype, you would test the genetics of her tumor to figure out, is she ER positive? Is she dependent on estrogen? Or is she not in terms of her tumor? And I just want to say this has been really powerful because for some women who we anticipate might be very sensitive to developing breast cancer, you can actually give these women tamoxifen preemptively as a preventative measure. So before they even get breast cancer, if a woman, because of her family history or her lifestyle or other factors, if a woman is very likely to develop a severe breast cancer, you can tell her to take tamoxifen early and she just will be less likely to develop it in her lifetime. So this is a very effective, and again, targeted therapy. It's not like there's no side effects, but the side effects are less severe compared to a treatment that literally targets all DNA. Are there any other questions up until this point? All right, so let's talk a bit about discovering new targeted therapies. And so I'm going to walk you through this diagram and explain what it's showing, but I'm going to just give you a couple of ways that we've sort of tried to discover new targeted therapies. Because again, you can imagine in the prior case with the woman who has breast cancer, if we test her cancer and we realize, oh, it's estrogen dependent, we've created this new option for her, right? She might be very responsive to tamoxifen, but imagine the patients who test negative. Like, what do we tell them? They basically don't have that option. It's harder to treat their cancer. So we're trying to think about what is true about their tumor that gives us a new way to treat them specifically that maybe isn't true of the women who use tamoxifen instead. So we're trying to discover these new targeted therapies. The ones involving hormones were a little easier because it leveraged what we already knew about how certain tissues work, especially those related to sex organs. But now everything else is sort of more of like this open search for anything that might be the Achilles heel of a given cancer. And so there's a couple ways we think about identifying new targeted therapies. So one thing I will mention is when we're thinking about targeted therapies, you can imagine, and I think I showed the inner ring last time in terms of a diagram, is that there's all of these facets of cancer. Part of cancer is that it is able to divide forever. Part of cancer is that it's able to invade and metastasize. Another part of cancer is that things that would normally trigger a healthy cell to die, such as breaking the rules or not enough food to go around, cancer cells have evolved to overcome that messaging. And so they're able to resist cell death. And so all of that inner ring is showing these traits that are true of cancer cells. And so what's being shown along the outer ring is showing examples of treatments that are basically trying to target that feature. So for example, if cancer cells are able to invade and metastasize, there might be an inhibitor of a specific protein or enzyme that has a role in cells' ability to move around, or cells' ability to go from one place and like land and survive that journey and land in another tissue. So what's being shown around the outer ring are examples of targeted drugs that are aimed at that trait. Like if cancer cells can change their metabolism, let's target their ability to change their metabolism. And so that's sort of one very targeted approach. Yeah, Sahil, that's a great question. Do we layer targeted drugs on top of each other? We do. We absolutely do. So one thing that might not have been obvious from that first slide I showed, here, actually, let me go back to it very briefly. From this sort of staging diagram and thinking about the treatments, something that might be not so obvious is that there are times where we kind of will try one treatment, and then when that doesn't work, we just go to the next one. So that's a lot of this sort of first line, second line is deciding, oh, we'll try this first. If that doesn't work, we'll go to the next option. But there are cases where we've done research and we realize certain things are really effective in conjunction with each other. And assuming that two drugs, which generally speaking then brings two different sets of side effects, if those still are tolerable, there are cases where we know that the combination is more effective. And we will actually talk about why we tend towards combinations. There's sort of like a theoretical reason why combinations do really work well that we'll talk about. But yeah, there are cases where we just sort of go from one to the next. And I think that tends to be where the two together are intolerable, or there's still this hope, oh, if this one really works, we don't even, like, we can save that other option till later so that we just have more options. So like, let's exhaust this one first. Yeah, but there is a lot of research where clinical trials are just basically like, let's try A plus B, or let's try A plus B plus C. That's definitely something that's always being researched. All right. I think I will actually skip this next one and go to here, because I think this is very relevant to how when you are someone who's very data oriented, I think this strategy of identifying targeted therapies might resonate a lot, which is to say, there's been a lot of work where people, as they are collecting all of these different tumor samples and sequencing all of them, they're really interested in, okay, well, what mutations are really common, either across all cancers or in certain types of cancer. So if you collected a lot of different lung tumors, what is like the general profile of a lung tumor? What tends to be mutated? And the idea is that if you identify certain things that tend to be mutated, you would think, okay, well then, is that a target? If there's something that's mutated and often is like acting abnormally in cancer, if I target it, specifically like the mutant or the overactive version, that might be an option for treatment for patients who test positive for that mutation as well. And I want to be clear, that's not true of all patients. So for example, here you can see this was true of like 13% of patients, but the idea is that you can imagine, oh, if a patient comes into the clinic, we might test for this one, and this one, and together that might account for 30 to 40 to 50% of patients have at least one of them. And so we might test them for all of these mutations to then point to, like, should you take drug A or drug B or drug C that target each of these mutations? And so there's been a lot of work where we've sequenced so many tumors to try to basically be able to develop, like, hey, a subset of lung cancer patients are eligible for this. We've developed a drug that now targets this. So we'll genotype them when they get diagnosed to point them to this therapy. So this is something that has happened a lot. As long as we've had sequencing available and been sequencing tumors, we've been working on trying to figure out what mutations are targets. But I wanted to be very clear, though, that all of these different options don't tend to work so well in practice. Generally speaking, once a patient is at a later stage, so the cancer has metastasized, I think I forgot to emphasize this, but when you're at a later stage in cancer and the cancer has spread, we often use a lot of these therapies, like these targeted therapies or chemotherapies, because the idea is that they go throughout the entire body, right? When you're a patient who's being given cisplatin, you'll get it usually for like an IV drip, or maybe you take a pill that sort of just goes throughout your whole body. It like it's flowing in your blood, going throughout your whole body. And the idea is you take those types of treatments, those systemic treatments, because your cancer spreads such that, yeah, we know that there's a tumor and we can, target that tumor, but the cancer cells could kind of be anywhere. And so you're taking the systemic treatment to hopefully kill the cells wherever they might be. Hopefully you'll hit the tumor cells wherever they have ended up. And so later stage patients, usually what we point them to is chemotherapy or a targeted therapy they might be eligible for, because it will go throughout their whole body. But again, these are patients who are at those later stages. Surgery was not a curative option for them, so we point them to chemo or we point them to targeted therapy. But I will just say, again, I mentioned this doesn't tend to be curative. It's more like it buys them more time. Generally speaking, we tend to see curves like this one. What this curve is showing you is sort of out of a total number of patients, how many were still alive after a certain point of time when they've been diagnosed, they've tried the localized treatments, and then they get on a targeted therapy or a chemotherapy. What you can see is that all of the curves collapse to zero, right? Ultimately, all of the patients in both cohorts died. But what happened to the patients who got the targeted therapy versus the ones who didn't is that it was shifted. So we've shifted the curve to the right, which just means that they get more time. They get some number of months, in lucky cases, they get some number of years where they continue to survive, but ultimately it doesn't cure them of their cancer. And I will just say that the closest we've come to that so far has been things like immunotherapy, which is where a lot of the excitement has come from. And so I'm just going to compare these two curves, which is on this side, it's as you've seen here, where it's like there's a shift. There's a shift, but ultimately both of them collapse to zero. And so in both cases, all patients die. It's just a matter of some of them get more time. What's being shown here on the right, and this is in the case of immunotherapy, is you don't necessarily see the shift, but there are a subset of patients. Again, what this is showing here is of the 100% of patients who were in the cohort originally. If all of them die, the curve collapses down to zero. But what you'll see here is that this curve actually doesn't collapse all the way to zero. So this is shown here in the purple. This doesn't collapse to zero. And so there's a subset of patients who just keep living. And so for them, essentially, they've been cured, or they're like they are sort of the exception versus everyone else who they had more time, but they still ended up dying. These patients just keep living. And it's not all of them. It doesn't work for all of them, but this in and of itself is just a change in what we've tended to expect, right? With every additional treatment, it's kind of been like, oh, I can shift it even more, but it still collapses to zero. That's been the dichotomy that we've been used to, is the original curve versus a shifted curve and how far we shift it, versus thinking about, are there patients that just keep living? So that's been a lot of the excitement around chemotherapy, but I did want to touch on why some patients, or why is it so hard to cure patients? Why is it that with all of these targeted therapies, ultimately all patients still die? Sorry, I should be clear. That's not the case in literally every cancer. There are some cancers where we can cure them with targeted therapy. It's just that tends to be the exception rather than the other way around. So rather than saying, oh, targeted therapies cure most patients, but there's some patients who it can't help, it tends to be that targeted therapies don't cure most patients, but there are a couple of exceptions where it does. And so some of you may have heard of, there's this drug called Gleevec, and it is specifically used in a type of blood cancer. I believe the cancer it's used in is CML. And so in that very particular type of cancer, for patients who are eligible for that targeted therapy, it actually works really, really well. Like a lot of those patients tend to be cured or survive many years. Some of them might relapse after a long period of time, but there are some patients who just seem to keep living with this drug. So I don't want to say like it literally never works. I would just say by and large, and especially for a lot of the most common types of cancer, like lung cancer or breast cancer or cancer of the brain, we tend to see this type of pattern more where, it helps for a while, but it ultimately can't cure them. And I just want to talk about why that is. And really, the theoretical point of view for why this is the case is actually evolution. So you may have learned about evolution in the context of like species of things like animals or plants or people or bacteria. And the idea is that with evolution, these different populations of living things, like I think the common example is like, oh, the gazelle, and the gazelle needs to outrun the lion. And the idea is, oh, the gazelle would evolve ways to be able to better elude the lion, right? So they can survive and they could reproduce. And so if there's a way they can evolve better camouflage, if they can evolve to run faster, all of those things would contribute to them being more likely to survive attacks by lion. And so their genetics sort of slowly evolve over time to select for, if a mutation arises that like leads to a coat that's harder for the lion to see, that gets selected for, and that becomes more common in the population of gazelle, because that gazelle basically lives whereas others might die, and they pass on that mutation. Cancer works very similarly in the sense of you have to think of it as a tumor is a collection of cells. Each cell has its own DNA. Granted, they all originated from your body, so they share a lot of DNA. But as cancers start to become cancerous, they're basically accumulating mutations, and each cell is its own agent. Like each cell may have come from another cell, but during the lifetime of that cell, they can continue to mutate because of, the cancer cells just make a lot of mistakes. The cancer cells are being exposed to all of these DNA mutating substances. And so each cell is kind of like its own agent that is undergoing changes. And if a mutation arises in one of those cells that is advantageous, it becomes selected for. So when we're thinking of a cancer, cancers arise from a single cell that accumulates some mutations and generally leads to that cell growing and dividing more than it's supposed to. And when it grows and divides more than it's supposed to, it ends up creating a population, right? Because that growth, all of the cells that descended from that growth, there's a collection of them, right? And each of those has a chance to mutate. So in this collection, there might be a certain cell that randomly gets a mutation that allows it to then do something that it was unable to do before. So for example, this could allow it to invade physically into a new space that it couldn't survive in before. And as it invades, it makes a home in that new space. It continues to grow and divide. And so it creates this population of cells, and each cell has a chance to divide, and each cell has a chance to mutate. And so if it mutates, it can gain a new ability, it can become selected for, and again, create its own new population. So it basically just continues to evolve from there. And that's what we actually see when we try to treat. So this TX basically stands for treatment. When we're treating, basically at that point in time, let me clear this. So when we're treating, at that point in time, we see this sort of collection of cells that makes up the tumor. And it's a very diverse collection, which is what's being shown with all these different colors. And when we're trying to treat them, we're trying to kill all of them. And maybe we're very successful at killing most of these, but maybe there is one cell that has a mutation that means it doesn't respond to the treatment. We've killed all of its competition. That cell is selected for. It is the one gazelle that outran the lion. And so left to its own devices, it basically keeps dividing, and all of its cells that it creates all carry that resistance mutation. And so that's what we tend to see in cancer, is that we'll treat with a targeted therapy. The tumor looks like it's responding because it'll generally stop growing. It might even regress. So the tumor looks like it's getting smaller and smaller. But then at some point, we sort of killed all of the cells that were sensitive to the drug, but there might be some number of cells that have a resistance mutation. And so at that point, they don't shrink any further, and actually they start to grow back because the drug makes no difference to them. They basically don't see or don't respond to the drug. So we see that with patients where the tumor regresses, stops responding, and then starts regrowing. And that is very typical because basically their cancer has evolved at that point. Are there any questions about that?
So my question is, again, refer back to the COVID situation. We know the RNA keeps on mutating.
Yeah.
So are we eventually going to face this kind of dilemma because of this mutation, the current vaccine may not work? So you mentioned about the shape of the virus may be the same, but who knows? I don't have any information to do any prediction.
Yeah, I mean, that's a really interesting question. I will say one thing to keep in mind is, generally speaking, the repertoire that a virus has to work with tends to be a bit smaller than a cancer cell, right? Like a cancer cell basically has all of the genes that you do and then starts to like use them in very creative ways, right? Like it takes your existing genes and maybe expresses them in new configurations or mutates them so it's expressing like a different version. Viruses have a much smaller genomes. So they, generally speaking, have a little less to work with. Like the amount they can mutate and kind of change what we're working with is smaller. I think what more people tend to be worried about and what leads to really big issues is when viruses sort of combine. So I'm trying to think of the picture. There's this really great picture of it. Actually, let me try to show it. So there's this really great picture. So when you're thinking about a virus, right? And I'm just going to draw like spikes on the outside because this is basically, the spikes are important because the spikes are basically how, if it runs into a human cell, it's basically looking for something on the human cell that it can grab onto and is the means by which it invades. So if this is a virus, if this protein is specific to this feature, and this tends to be the case for COVID, that it's something that's common in your lungs, it basically can bind to that, and that's its entry route, is that it binds to those types of cells and it enters, right? So someone who's getting infected by a virus, it's like, oh, just having the virus interact with your skin, for example, isn't enough because the thing it binds to isn't on your skin. It's a specific feature on your cells that are common in your lungs or in your like the tract leading to your lungs. So what I'll say is that the amount of genetics that's in here tends to be pretty limited for viruses compared to cells. So first off, that just means like probability wise, if you're dealing with something, I don't actually know how long the COVID genome is, like just compared to a human where it's like, oh, we have, 3 billion bases to work with. It's much shorter for, actually, let me look it up. COVID genome length. It is 30,000, 30K. So compared to 3 billion, it's a lot smaller. Mm-hmm. And so the amount that it can mutate tends to be quite limited. Like it doesn't sort of have as many of the endless possibilities for what it can do. I think the thing to also consider is that I think there's a lot of concern around it mutating these outer proteins so that what our immune cells, right? Our immune cells are supposed to develop these receptors that can find these viruses. Mm-hmm. And the idea is if they change their spots, then our immune cells can't do that anymore. But I think a thing to keep in mind is these outer proteins are serving a purpose for the virus. Like it literally can't change its spots to be any color, any shape, because at some point, if it changes too much, it actually can't bind to the lung cells anymore. Sorry, apologies if you can hear that. So I think that's a problem for the virus where it's like there are some mutations that might make it really good at evading our vaccine immune response, but that also might make it worse at infecting, right? Or it might change who it infects. I mean, I think that might be something to be concerned about, is that if it somehow very effectively changes who it infects, it just means that yes, our vaccine won't work as well, but I think also then the the disease it leads to will be different, right? Like the disease
Mhm.
we know is because it infects lung cells. So if
Yes.
that changes its target, it's going to be a different disease in some sense.
Mhm.
So that's what I would keep in mind, is just it's smaller. It doesn't have like the potential to mutate literally in any way, because it's kind of facing this trade-off.
Mhm.
It's like, oh, if it evades the immune destruction, it just might also not be as good at being a virus. Those are the things I would consider. I think one of the concerns people have had is what it's something that happened with I believe it's what happened with the 1918 flu, which is the concern of a co-infection that leads to exchange between two viruses. So generally speaking, when a human cell or an animal cell, is infected by two different virus particles at the same time, what can happen is the way these viruses often work is that they kind of just basically inject their genetic material into the cell and make the cell do what it wants. And often what it wants is it wants the cell to become a factory to make more virus. But if you have a co-infection, so two different viruses at the same time, their DNA both gets plopped in here. And in generally speaking, it's not like it knows which DNA came from who. And so what can happen is you can end up with a virus that's kind of like a hybrid between these two viruses. And I think that's something that people are concerned about with viruses in general, not specific to COVID, just in general. Usually this hybrid virus can sometimes be the worst of both worlds. So I think that's something that people might be concerned about. But generally speaking, like we deal with this with the flu every year, is that it changes its spots enough that it avoids our vaccine response. But I think there's only so much it can change that, like, I just don't know if COVID variant in future years, I don't know if we'll have endless variants, or even then, I don't know if the variants will just keep getting worse. They just might be different. Yeah, I'm sorry. I think it's very hard to say, but those are just some of my early thoughts.
Thank you so much.
Yeah, no problem. So let me see. Sorry, I'm just looking ahead at stuff. I think this is a good place for just one. Well, actually, we go to 10, right? Is that accurate, Edward? Oh, you're muted.
We ended upon 9:50, but we can go till 10.
Oh, no. I don't believe in keeping people beyond. So I'm trying to think about what would be more interesting for people to hear about. Are people more interested in hearing about immunotherapy, or do they just want sort of an example of algorithms being used to guide treatments? People can sort of either weigh in verbally, or they can weigh in the chat what they'd be more interested in: immunotherapy or applications of ML in treatments. All right, I'm hearing applications of ML. Okay, so let me give one example that was actually research I worked on. So let's go to here. All right, so let's talk a little bit about examples of this kind of research. So again, we face this really difficult issue where we have patients, we're trying to figure out what therapies would work better in them. Often better just means longer survival times. It doesn't often mean cures, but even then we still think that's a worthwhile area to pursue. And so I have several examples that are shown in my slides, but I'm actually going to focus on the one that I worked on. So the one that I worked on, one thing I have to establish about the thing that I worked on is that we are dealing with sort of two levels of data. In this data set, what we have is, like, first off, what we have is basically a collection of cancer cells. But what I'll tell you is that what can be the case for this type of data is that we're collecting single cell data where we basically measure and profile each individual cell. But what you can imagine is when you're doing research on patients, you can have this data collected from every single patient that you work with. And so you have kind of two levels of data. You have the level of the data that's like each individual patient, but then if you're collecting a sample from them and characterizing their sample with single cell data, you actually get two levels of individual. There's the individual cell that came from an individual patient. So there's a population of patients, and for each patient, they have their own population of cells that we're working with. And so that's something that leads to a lot of interesting, like, multi-level problem solving, where you're like, oh, I'm trying to predict something for the patient on the basis of the diversity of cancer cells they have. So if you're used to working with data where it's like, oh, for every single patient, I have these rows, and on the columns, I have different features about them, it actually becomes that this is a tabular data set you have for every single person. Each person has their own tabular data set for every cell that you collected from them. And so again, this is like a multi-level problem solving step. But I just want to give you a glimpse into how we deal with the single cell data. So just like ignoring the individual that it came from, let's just focus on these single cells. So I was working in a lab where we had a means by which we could take these cells and sample them and basically measure multiple features in each individual cell. We were measuring it on the protein level. So again, we talked last time about like measuring their genetics, measuring what their RNA they're producing, and then measuring the protein they've produced. We were measuring protein they've produced. So we took these cells, and what we did is that we exposed them to a couple different treatments that are very commonly used to treat this particular type of cancer. So the cancer I was dealing with is called multiple myeloma. It is a cancer of the blood. So you would collect cells from their blood or from their bone marrow. And so we take these cells and we expose them to the most common treatments that are used for this particular type of cancer, because our interest was looking at different time points, like before treatment, during treatment. Sorry, I'm gonna get rid of my little doodle. And after treatment, we wanted to see, okay, what's happening? Like starting cells in progress of being exposed to drug and, responding to that drug, and then cells at the very end where it's kind of very clear that some cells have died and some cells have lived. And so those living cells are resistant for some reason or another. And so we had a means by which we could collect cells at these different time points and measure up to 40 proteins in each individual cell. So again, the data format was like each row was a different cell collected at a different time point. So you had to remember each cell, which time point it came from. And then on the columns was like amount of protein A, amount of protein B, amount of protein C, and so forth. So that's the data set we collected. You can imagine it's like a tabular data set of cells, maybe for each separate time point, and each separate treatment as well. And what I was interested in doing, again, methods wise, it wasn't the most like sophisticated method, but again, we're always dealing with this trade-off where it's like the more complex the method is, the more we don't actually understand what is happening under the hood. So the way we worked with this was actually, we wanted to do a classification, which was to sort of determine what cells are ultimately the ones that die with drug, which is the sensitive cell, versus the cells that don't die with drug, those are the resistant cells. So if you have this data where you've treated them with drug and some of the cells are alive and some of the cells are dead after drug treatment, so again, resistant versus sensitive, we wanted to use a classification model to figure out, well, what are these features that lead to cells that tend to be alive or tend to be dead at the last time point. And so we were interested in basically identifying the most strong features that lead to cells being classified as sensitive versus resistant. So if you're not familiar with how random forest models work, you don't get one single tree. So I've drawn a single tree here for the classification where it's like, oh, depending on the levels of this protein, you would go into this bucket versus this bucket. When you're dealing with a random forest, it's a forest. So there's too many of those trees. You don't have one single tree or one consensus tree, but you do get a sense of what tends to be an effective split I wrote gate here because that's the word we use a lot in biology, is the gating based on expression, like high expression versus low expression of a protein. But you can think of it as like the split in each level of the tree. So something that tends to pop up a lot, like let's say marker X does a really good job of splitting between cells that are resistant versus cells that are sensitive. In a random forest model, you can see what tends to be a good splitter, and those become like more important variables. So they're weighted more highly. And so even though you don't get a single tree, you do get that information. And so we got that information, and what we saw was that there were clearly a most important marker for distinguishing cells that live and cells that die. And it actually tended to be pretty similar between these two drugs that work in wildly different ways. Like the biology of what these two drugs are doing is incredibly different. And yet the same two features were the most powerful for making the cells more likely to be resistant or more likely to be sensitive to both of these drugs. And so again, if you're thinking about the goal of this project is to identify what makes a cell more likely to be resistant, and ultimately the practical application of that is if you know what makes that cell special from the get-go, like if you know, oh, ultimately this patient will probably relapse because there are these cells that have a lot of these proteins and that for some reason makes them more hardy, like more resistant. If you know that ahead of time, your goal would be, oh, can we like preempt that somehow, right? If we can kind of cut them off at the pass, we know that those cells will ultimately probably not respond to the drug. Can we add something additional so that they'll kind of, like, we hit most of them with the first drug, but the second drug kind of hits the ones who would have responded or would have failed to respond the first time. We still eliminate them with the second like line of attack. And so that's what we decided to pursue, which was that we had a specific therapy against MCL1, which is one of these two top features. And so what we did is we showed that if you co-treat with a therapy against MCL1, so that's what's actually shown here on the far right, if you co-treat with them, they're very synergistic. And I want to be clear to define what synergistic means for cancer biology. What synergistic means for cancer biology is that the effects of the drugs are not additive. So if one drug kills 10% and another drug kills 30%, it's not like you just add those percentages together. It's actually that you kill more than you would have expected than if you just added the two together. That's when it's synergistic. And what we found was that using this inhibitor against MCL1, which was that highly predictive feature, using that in concert with the original drug was very synergistic. It was like we killed the original cells with dexamethasone, and we killed some cells that would have been left over with the MCL1 inhibitor, but we also just generally killed more than just the two of them added together. I just wanted to say that these other two were just other examples of treatments that are kind of similar to MCL1, and they kind of help, but they don't help to the same extent as MCL1, because that was what we identified as the key feature of our resistant cells. And I just want to share, we did find that that ultimately was true of patients as well, is that patients just did better with the two treatments as opposed to with any treatment alone. Like we had more killing of cancer cells. But I do want to really highlight again, and this is a continuous problem, this middle bar here, these middle samples, these dots are showing for each patient how many cancer cells did we kill with the combined treatment. And you can see that each point tends to be lower than either treatment alone. So these are just one treatment at a time. And you can see, for example, this purple dot, this treatment actually led to more cells being alive, which was interesting, versus this treatment killed some of the cells. But with the two together, we tended to kill more cells, but it also tended to be more than just the addition. It would kill like multiplicatively. But I do want to point out that most of these don't actually go to zero. And I think that's a very important thing to highlight, is that we, again, kill more cancer cells, but we don't usually kill all of the cancer cells. And that's where we draw the line between being curative versus just helping prolong life.
Mhm.
All right, I think realistically, that's all I had time to talk about. I thought I had less to talk about, but I didn't. So I think I wanted to just put this slide up and just like open it up for questions, broader questions. I just wanted to put this here that if you're interested in careers that relate to cancer biology and AI applications, there are a lot of options. So I just put a couple of different companies or nonprofit institutes that I know have actively recruited a lot of computational biologists recently, or computational folks who work on biology problems. There's a lot of issues around, again, trying to, create the best algorithm to know how to stratify patients or just be able to dive into the data and better understand how cancer functions or a specific type of cancer functions. All of those are really active areas of research. And of course, in current USA, I know that COVID is dominating health interests right now, but I will say that's kind of anomalous. Like for so long, America has been sort of this first world country where we don't think a lot about infectious disease, with like a couple of exceptions. Cancer has by and large been like the thing that dominates the American consciousness in terms of health. And we've just kind of been in a weird time where that's actually not true, but that's been true for like decades. Like I think the war on cancer started in like 1950, and we've been fighting it ever since to try to understand how to cure cancer. So I think this is a very active area if you're interested in it. And it really draws on a lot of different understandings of modeling and the different types of data you'll work with.
Yeah, definitely. This is a very critical area. So one quick question I have is, one of my colleagues, Jeff Huber, started a company called GRAIL, GRAIL. The mission was to detect cancer early. So do you know anything about the work at GRAIL?
I'm not familiar with it.
I see.
Sorry, I'm
Okay.
just like looking it up.
Yeah. Yeah, to be able to detect early seems to be very useful, right? Like in my age, I will be very happy to be able to do some kind of early detection.
Yeah, that's a great point. I didn't even bring it up, but there's an entire world of cancer screening, which we consider different. Like cancer diagnosis versus cancer screening. Cancer diagnosis is where you have a reason to suspect there's cancer, right? Like you're like, oh, you have symptoms, like you're coming into the doctor's office for a reason. And so the issue of diagnosis is like, yeah, you don't want to misdiagnose. You want to understand what's happening as best as you can. That involves staging or like how aggressive the cancer is, or if it's a certain subtype. So that's diagnosis. Screening is where you find cancer where you have no reason to suspect the person has cancer. And so that actually usually tends to be imaging related things. So like for women, it's like mammograms. For general population, like colonoscopies. Like there's lots of ways we try to screen for cancer ahead of time, but a lot of them are intense, like resource intensive, and a lot of them are imaging based. So I do know, I'm just looking at GRAIL, it's like there is a lot of interest to see if you could detect the hints of cancer from a blood sample. Like taking blood and testing blood is like one of the most like common workflows in healthcare. So if you could just like take blood and you're like, yeah, I'm going to test your cholesterol, but I'm also going to like see if you have cancer. Like that would be very easy to integrate into healthcare pipeline. Like it wouldn't be as onerous as currently. We just have to bring people in and do a colonoscopy, which takes a lot of resources. So I know there is a lot of interest there. I think a lot of times, though, what I imagine for those types of problems and trying to solve those problems is it's partially a hardware issue. Like it's partially like, do we have technology that's sensitive enough to be able to pick up on the signal? And then on the software side, it's like, okay, if we have this really dim signal and it's a mishmash of like, it's very noisy, can we still tell that it's something significant? So I think that's still just an active area, both on the engineering side and also on the data side.
Okay, thank you.
Are there any other questions I can answer? Do blood tests currently screen for blood cancer? I don't know how routine it is. It is something that tends to be very obvious. If someone has a blood-based cancer, if you take a blood test, it both can affect the way your blood behaves. So for example, the way that you form like clots, like if you bleed, like the ability of the blood to clot the way that we'd expect it to is changed. So a nurse might be like, wow, this blood is behaving kind of abnormally. But also if they do any kind of test that involves a smear where they just like splay out the blood and they look under a microscope, if you have a blood cancer, they would be able to see that right away. I just don't know that every single type of test involves looking under the microscope, but those tend to be really obvious. Yeah, blood cancers though tend to be very, like they tend to manifest quickly and have symptoms quickly. Yeah, I think health and physical examinations, I think that's really interesting because again, things like colonoscopies or prostate exams can help with early detection, but like it's a very private experience. And so a lot of people just don't want to get screened. Like they're like, oh, I don't want cancer, but I also either like it's a lot of resources, you need good healthcare access, but a lot of people also just don't want to go get the screenings done because they're very invasive or if they feel personally invasive. So that's also an issue as well. So like something like a blood test, I think is very normal to people. And I think they would be more willing to be part of that procedure. Yeah, I don't know if there's ways to detect other types of cancer from blood imaging. Technically, if you have a late stage cancer, one way that cancer might be spreading from one place to another is through the blood, but it's very rare. I mean, it's very rare to see, right? Like, so if you have a cancer that started in one organ and is metastasizing to another organ, and the way it's doing so is by transit through the blood. So technically in your blood somewhere, there might be transiting cancer cells. I just think that the chances that you'll like extract some blood, look under a microscope and see that cancer cell is still fairly rare. Like there's so many blood cells, like you've kind of drowned out any of these rare spreading cancer cells. But yes, technically there are these things called circulating tumor cells. So the common name is CTCs. And there's been interest in either finding them, but again, they're very rare. So it's hard for researchers to specifically get them, even if they're trying to pick them up. And so that's not even to diagnose, like you already know the person has cancer and you're just trying to catch them. That's very challenging. But I think there is interest, again, it kind of hints at this company Grail is that when you have CTCs, like these circulating tumor cells in the blood, they very often break, like they very often will just die in transit because they're undergoing so much like pressure, like the blood flows through fast and at such high pressure that cells often will break apart if they're not blood cells. Blood cells are designed to do it, but you know, a cell from your lungs isn't designed to withstand that type of pressure. And so those cells will break open and their DNA and their contents will then be flying around in your blood. And so a company like Grail is trying to find the fragments of a cancer cell in your blood. That's a little more likely that you might like pick up on, hey, there's some rogue DNA in this blood sample. Like that is something you might be looking for instead, rather than imaging and seeing a cancer cell. What am I most excited about? I do think immunotherapy is very exciting. As I said, I mean, my thesis work was on targeted therapies, but it was kind of disheartening because like the evolution paradigm is so powerful, right? Like the diversity of life we see on this planet is because of evolution, and the fact that you can see such clever evolution, like it's supposed to be a completely random process, and yet you see living things adapting to the most ridiculous circumstances on this earth, like evolves to eat this one thing and live in this one place and adapt to this one crazy weather pattern. And so knowing that that's the power of evolution and that cancer cells are really in tune with that power is a little disheartening when you work on targeted therapy because you're kind of very outpaced in terms of what you do, versus immunotherapy is using your cells to fight these cancer cells. Like your cells unfortunately have kind of been suppressed during cancer, like for some reason the cancer has evaded your immune system. But if you could empower your immune systems are a little more flexible or adaptable than like a chemical compound. And so I think that's exciting if we can somehow empower our own immune system, because our own immune system is flexible. It's not just sort of, do we see compound or not? Does compound bind to target or not? There's like more to it. Yeah, so I think that's what I'm most excited about. Yeah, any other questions?
Okay, so if you have more questions to ask Melissa, just enter to Slack or just send email to Melissa directly. I really like to appreciate the insightful and edifying lectures given by Melissa, and it's always my pleasure to listen to your lectures. Even this is the third time. Every time I can learn something from you, it's really wonderful. So
thank you for having me.
Appreciate very much. Yes.
Yeah. Awesome. Thank you. Yeah, feel free to be in touch. I put my email in the chat.